Hello everyone. This is Ercument Cicek. I'm a PI at Bilkent University in Turkey. I'm going to talk about our language model to design protein binding RNA. As we have seen through the talks today, protein and RNA interactions are quite important for various biological functions. And RNA is an alternative to ligand-based drugs is a promising direction because only 15% of the proteins have binding pockets for ligands. And in other contexts, RNA-based drugs are favorable because they don't trigger the immune system as much and no genotoxicity risk and also swift production once the chemistry is set. So the problem we are facing is designing an RNA sequence that strongly binds to a given target, and that's a protein. So the challenge is the sheer number of possibilities in the design. Unfortunately, current practice in the lab is through trial and error. So either you select the library and eliminate it through experiments, or if you can find a similar solution to a similar problem, then you just re-engineer the solution. But however, these take time, cost money, and prohibit novel design. So computational methods might help, but due to critical limitations, they are only proof of concept currently. These mostly rely on generative models, which train on natural sequence or structure RNA data. And they first learn to generate RNA, and then in the second step, they are optimized to bind to the target. So the state of the art in RNA sequence design has a few works. I'm showing you a picture from our earlier study where we first learned to generate RNAs using a GAN-based architecture. And then the frozen generator is taken and it generates some candidates, which are then optimized using some binding predictor. So given a target, it predicts if it's going to bind to this protein or not. And so based on this feedback, the generated sequence is optimized. However, this worked. So we designed an RNA to bind to SOX2 protein and validated it in silico and also in vitro. But this approach has critical limitations. The first is learning to generate RNA. So this latent space is only based on RNA. It doesn't know about the interaction at all. So it doesn't know about proteins. The second and maybe more important bottleneck is that this information on binding, these, in this case, deep bind models, are available only for 700 or so proteins. So it means it's not possible to design for novel or new proteins, and most of the proteins actually. So the state of the art in RNA structure design has, again, a few models. They mostly rely on a diffusion model, which is conditioned on a protein. So the model learns to denoise this structure and learn to generate a new novel backbone, which is then inverse folded to generate the sequence. Now, the bottleneck here is the lack of ground truth structure. Last time I checked, the RNA solo database had around 10,000 ground truth structures, and that depends on how you define a ground truth. So this is order of magnitude less than the available structure data for proteins, and also RNA structure is much more flexible than protein structure. So we have a harder problem and less data. This is also admitted in these two works. So for instance, the generated backbones closely match the already existing structures in PDB, and for instance, RNA flow doesn't outperform random structures or sequences when the RNA is conformationally diverse. So that's why we think this is a huge bottleneck, and we rely on sequence design. And here is the idea. So when we have ChatGPT, we can just input an English sentence and it can translate to French. So in RNAtranslator, we are going to input the target protein of interest, and it's going to generate a novel RNA molecule that would bind with it. So in ChatGPT, we have input translating to the output. In RNAtranslator, we have input binding with the output. So here we are not trying to design the RNA and then optimize it as a post-process. Here we are trying to model the interaction. So hopefully this is going to help us learn about the protein as well and design a better RNA. And technically we are not limited by these guidance models. So we can virtually design for any target protein sequence. So this is our model. It's trained on 26 million protein RNA interactions from RNAinter database. So the protein goes into, so this is a transformer-based architecture. the protein is encoded, and the RNA is decoded, conditioned on the encoded protein embedding. And we generate the RNA, the binding RNA, with next token prediction. Then this architecture is fine-tuned based on more reliable data, which are based on clip experiments to have better performance. And at the time of inference, we just input the protein, and the RNA is sampled from the latent space. How do we evaluate? So for a target protein X, we train a deep clip model using the ClipDB dataset. So once it's trained, we input the generated RNA and estimate the binding affinity. We also have a left out ground truth data for this protein, so we can see how well we have done. We also do some molecular dynamics simulations. So we predict the structure of the RNA, we dock it with the protein of interest, and then do molecular dynamics simulations. So we have numerous benchmarks. So we show that we can generate natural-like RNAs with high binding affinity to target and better than existing methods. But I don't have time to go into those. I refer you to our preprint over here. But I'm going to show you a couple of results. So we design. So these are not even on the preprint. We designed an RNA to bind to a therapeutic target, which is thrombin in this case. So it's a target to treat thrombosis. So this is the result of our design versus a natural random RNA, so we don't expect it to bind, and then a natural non-thrombin binding RNA. So here you see the hydrogen bonds and the binding energy. So these are the distribution of the validated RNAs. So we expect ours to form more hydrogen bonds. And this is the binding energy, so the lower is better. So we expect our generated RNAs to have better structure, so more stable. And this is our top design, which this is the result of the simulation. Now, this is, I think, more interesting. Here, I am showing you our design for a novel protein. Here, by novel, I mean, so this is PRP4K, it's a kinase, and we don't have any interaction data in the databases for this. So currently, it's not possible to design a binding RNA for this protein. So for that reason, we cannot compare with the natural binding RNA, we can only do simulations. So it's our design versus a random natural RNA, which is not expected to bind. So here you see the number of contacts through the simulation and the number of hydrogen bonds. And here you see the binding energy through time simulation. As you see, it's much better than the random RNA. And here you see the free energy landscapes. Here what we expect, so the stable binding would give us a narrower landscape and a deeper basin. So here, as you see, this is our designs. It is a much deeper, it's deeper than the random natural RNA. And as you see, it's covering a larger region. And this is the expected structure of the complex. So with that, I would like to thank you. This is my team, for former and current students. Here is our preprint, and I can take questions.
Maybe one short question.
Hi, very inspiring talk, like to design a novel RNA using a translation ideas. I'm just wondering, do you have any kind of negative samples? For example, if there was a protein could never bind to any nucleic acid, and then I input that sequence in your model, what it will predict?
So it's a protein which cannot be bound by any RNA.
Yes, for example,
so if
cannot
what
be
would
bound
be this? Yeah, we haven't tested, but I don't know any proteins that we know of for sure not be bound by anything. But if we did, we could test it. It's an interesting question.
Well, thank you very much.